Bukan siapa ke?
Jana.
Cikgu dah boleh mula
Mula kita boleh mula dah. Okay, jom.
Okay, Jana mula dulu.
Okay. Siapa fokus?
Boleh buka nota kalau boleh. Open your notes. Page 95.
Semua dah masuk ke ni? Afrina, Dashiga, Dinesh, Farid, Ikram, Khairina, Nadira, Jiha, Fahana, Risha, Bihang, Syauki. Yang 3 orang yang live CID ni Farah Cik Farah.
Mana yang lain?
Okay,
Nampak lagi tak? Nampak kan?
Nampak.
Okay.
Kawan lain nampak tak slide Farid?
Nampak.
Okay.
Okay, Jannah dan the gang boleh start.
Okay. Mana? Okay. So enzyme, okay. DNA replication dia akan ada 7 enzim yang involved.
Cuba
First is DNA helicase.
Jana play Jana.
Apa?
Slide tu Jana play kan.
Play kan Farid.
Okay, jap. Okay. Okay, teacher. Tak ada
Tak ada pun.
Sekejap
Semula.
Bentar.
Jana dengan Nat Okay.
Nampak tak?
Okay, dah.
Nampak.
Okay, Farid, nice.
Okay.
So,
Okay,
okay. Bismillahirrahmanirrahim. Assalamualaikum. Saya Jana. Okay, so hari ni kita nak cakap pasal DNA replication. So DNA replication ada 7 enzim yang akan involve. First is DNA helicase, second topoisomerase ataupun DNA gyrase. Yang ketiga adalah single stranded binding protein which is SSB protein. Yang keempat primase, DNA polymerase 3 and DNA polymerase 1. Akhir sekali is DNA ligase. Okay, SSB protein is part of the enzyme sebab protein adalah enzim. So kita ada 7 enzim yang terlibat dalam DNA replication. So enzim yang saya tulis ni dia ikut DNA replication punya step. Dah siap-siap ikut tau. So korang kena ikut benda tu. So kalau ikut benda tu korang tahulah which one untuk apa. So senang korang nak ikut function dia semua. Okay, so first DNA helicase tu function dia adalah untuk unwind the double strand DNA untuk form replication fork. Replication fork ni terbuka dua. sebenarnya dia terbahagi kepada dua. Macam
Nak
ni.
tunjuk gambar tak? Nak tunjuk gambar tak?
Boleh juga.
Tunjuk gambar. Saya tunjuk gambar.
Ke nak saya buat secara manual?
Boleh je kot.
Okay. Dira, kau punya kamera. Semua buka Dira punya. Macam nak buka yang jadi ke depan? Okay. So yang ni, saya tengok kat kamera Dira. Yang ni, dia ada, yang ni adalah replication fault which is dia terbahagi kepada dua. DNA dia
Tunjuk kat mana Jana?
kat mana? Kejap dulu.
Tunjuk macam tu.
Jana perlu pertolongan?
Okay, kita sambung. Okay, dah boleh situ. Yang ni, collect. Cuba tengok saya punya gambar. Okay. So,
Tunjuk kat mana Jana?
yang dekat saya.
Jana punya slide, screen.
Jangan saya punya screen.
Sikit, Jana kena share. Jana yang mana? L mana ni?
Dia guna kamera je cikgu.
Oh, orang lain nampak tak? Kawan lain boleh nampak tak?
No.
Nampak cikgu.
tak
Tak nampak.
kan? Nampak tak?
Okay, sekejap.
Tadi saya nampak, saya tak nampak lah.
Tak apa, Jana sambung dulu. Gambar tu nanti later.
Gambar last.
Oh ni gambar ni. Okay.
Okey dah.
Okay nampak?
Korang nampak kan yang mula-mula DNA tu dia double stranded semua. Lepas tu dia terbahagi kepada dua. Itu adalah replication fault.
Gambar Jana gambar Jana tak clear Jana. Tadi nampak betul tak clear.
Macam mana ni?
Tak apa Jana, continue dulu present. Nanti later kita discuss, okay? We dah start dengan okay lah tu. You introduce all the 7 or the 6 enzymes, okay? Later on we summary with the diagram. So you proceed dulu dengan presentation.
Okay.
You just deliver your content dulu dan kita guna to recap, to summary, then kita akan go by diagram
Okay.
Alright.
So kan DNA helicase tu untuk unwinding the double strand DNA untuk form replication fault.
Okay, so Farid, on balikkan your slide, Farid.
Okay, kejap
So korang imagine je, DNA kan ada double strand. Lepas tu bila korang imagine double strand tu terbahagi kepada dua, dia terbuka. So itu adalah replication fault. And dia akan produce 2 single strand of DNA by breaking the hydrogen bond. Okay. Second enzyme is topoisomerase. Topoisomerase to relieve tension of the superchiral DNA molecule by nicking and cutting certain place on the phosphate backbone. So korang akan nampak ni masa dia orang explain dekat DNA replication tu. Korang akan tahu macam mana dia orang akan nicking and cutting. Lepas tu yang ketiga is single stranded binding protein ataupun SSB protein. SSB protein ni untuk stabilize the unwound single DNA strand. And okay, Dira. Okay, sekejap Dira pen. Bukan, nak jatuh. Sambung.
Yang keempat, enzim primase. Dia punya function is to synthesize the short RNA primer by adding free RNA nucleotide. And then DNA polymerase 3, it function to adding free DNA nucleotide from 5' to 3' and of RNA primer. So DNA polymerase 1 is to remove the RNA primer and fill the gap with DNA nucleotide. And the last is DNA ligase. Joining the Okazaki fragment by forming phosphodiester bond to form continuous DNA strands. So that's all.
So senang cerita, korang kena tahu dulu semua enzim function dia untuk korang kena tahu enzim dengan function ni. So korang akan lagi senang nak faham macam mana DNA replication tu berlaku. So okay, seterusnya.
Mana Farah?
Saya. Mana ni? Farid ada gambar tak?
Gambar last. Kena guna gambar tanpa slide.
Saya nak
Farah.
Guna gambar
Farah.
Sekejap Kita guna gambar kejap.
Tak apa, saya nak tengok. Saya nak baca.
Nampak tak? Nampak tak gambar tu?
Okey. Nampak? Cikgu nampak
Nampak,
Farid?
Tak
Orang lain nampak tak?
Saya nak besarkan.
Besarkan sikit.
Orang lain nampak tak nampak? Ikram nampak?
Semua nampak
Nampak cikgu?
Okay, good.
Okay. So, ni step untuk macam mana DNA replication tu.
Buat sikit suara Farah.
Hello. Dengar tak semua?
Farah
Dengar?
kurang jelas.
Louder.
So, step 1. Unwinding the double helix DNA strand. First, kan DNA helicase. So, orang tengok kat situ DNA helicase kan? Dia unwind the parental double helix. So, first kali DNA helicase ni dia akan bind dekat origin of the replication. Origin of replication. Kayaknya kat awal-awal DNA helix tu dia akan bind kat situ untuk pecahkan hydrogen bond. Bila dia pecahkan hydrogen bond, dia akan hasilkan replication fork. Replication fork ni yang bentuk Y tu. Dia macam terbuka kan. Tu ialah replication fork. Faham tak? Okay, and then each DNA strand ni dia akan act as a template. Maksudnya sebagai acuan untuk DNA baru. Untuk apa kita panggil? New DNA strand. Okay, and then single stranded binding. Okay, maaf bising. Okay, and then single stranded binding protein ni dia will stabilize the unwound DNA strand to prevent reformation of double helix DNA. Okay. Single stranded binding protein ni dia akan bind dekat single strand, dua-dua single strand tu near replication fork. Maknanya belakang DNA helicase. Function dia bind dekat situ supaya tidak berlakunya pencantuman information cantum balik double helix tu. Dia akan jadi double helix. So dia tak nak benda tu, sebab itulah single stranded binding protein akan bind dekat situ. Faham? Okay, and then topoisomerase pula. Betul. Topoisomerase ni dia akan bind dekat depan DNA helicase. Korang tengok gambar tu kan, DNA helicase akan ada topoisomerase. So dekat situ function topoisomerase ni bind dekat depan DNA helicase untuk prevent supercoiling. Kan dia nak breaking down the hydrogen bond dekat DNA helix. So bila dia buka, yang belakang DNA helix tu dia akan berpusing. Dia akan tak stabil. So to prevent supercoiling atau stress dekat double helix tu, sebab tu lah topoisomerase akan bind dekat depan DNA helicase. Seterusnya primase. Primase dia akan bind dekat 3 end of the template. Kau boleh tengok kat gambar tu kan. Mana primase? So primase ni dia akan form primer. Primer ni dia is a short sequence of RNA. Dia akan initiate DNA replication process. So barulah replication process tu berlaku dengan DNA polymerase 3 akan bind dekat depan primase. Adik?
Dah
Dah. Nak saya habiskan ke?
Tak apa, aku sambung sikit. Actually untuk DNA replication ni, jenis DNA yang kita gunakan ialah kita guna semi-conservative replication. Model. Actually ada 3 possible model for DNA replication. For DNA replication, model yang digunakan ialah semi-conservative. Sebab semi-conservative replication ni, semi-conservative model ni akan produce molecule of DNA with both old and new DNA, but each molecule will be composed of one old strand and one new strand. So dia akan hasilkan DNA, dia akan ada DNA yang lama dan dia akan hasilkan DNA yang baru. So dia akan hasilkan banyak DNA lah. And then itu ialah salah satu untuk rules in DNA replication, semi-conservative model. And then untuk start DNA replication ni mesti synthesis always in the 5' to 3'. Nampak tak kat sini? 5' to 3'. And then dia akan, yang bawah dia akan 5' to 3'. Nampak tak?
Okay.
And then dia akan so DNA dia mesti kena complementary base pairing. Kita yang ni gasity. So A will complementary with T and C will complementary with G. Okay. And then sambung balik, yang first step tadi ialah unwinding the double helix. Ini. So itu first step. DNA yang digunakan enzim yang digunakan dalam first step ada 3. DNA helicase, single stranded binding protein dengan topoisomerase. Itu ialah enzim yang digunakan dalam first step. And then second step is synthesis of RNA primer. So enzim yang digunakan dalam dalam second step is DNA primase. So that's why DNA primase synthesis RNA primer to initiate the process of replication by adding RNA. So second step ni dia akan iaitu DNA primase ni akan memulakan proses replication ni dengan menambah adding RNA nucleotide yang ini. RNA nucleotide using 3 end of parental DNA strand as a template ini. Okay. Then the new DNA strand will start synthesized from the 3 end the RNA primer. Ni RNA primer. Yang warna hijau ni. Yang warna merah ni RNA primer. Yang tu primase, ini RNA primer. Okay. Third step is assembling complementary strands. Enzim yang terlibat dalam assembling third step ni ialah DNA polymerase 3. DNA polymerase yang ni. Nampak tak DNA polymerase 3? Okay, DNA polymerase 3 ni apa yang dia buat ialah dia akan tambah free DNA nucleotide. Ingat tu, free DNA nucleotide. Actually dari mana datangnya free DNA nucleotide tu? Daripada makanan kita. Apa yang kita makan tu ialah dia akan jadi free DNA nucleotide. So dia akan add free DNA nucleotide to the 3 end of RNA primer and complementary to the basis of the template. Ini. Okay. And then after that this strand grows in the direction 5' to 3'. Yang ini. Nampak tak yang warna hijau ni? 5' to 3'. Since nucleotide are added only to the 3' end of the growing strand. Faham tak step 3 ni? Assembling complementary strands. Faham Okay. Step yang keempat ialah formation of leading and lagging strand. Okay. Dia ada dua strands iaitu leading and lagging strand. Okay, boleh tengok kat sini yang nombor 3 ni, yang warna hijau ni ialah leading strands. Leading strand ni ialah dia towards replication fork. Dia punya arah dia towards replication fork. So dia function ialah the leading strand is synthesized continuously in the 5' to 3' direction by DNA polymerase. DNA polymerase. And then satu lagi strand iaitu the lagging strand. Lagging strand ni ialah against replication fork. Dia again replication forks. So the lagging strand is synthesized discontinuously. So primase synthesize a short RNA primer which is extended by DNA polymerase to form an Okazaki fragment. Itu untuk step yang keempat. Step keempat ada dua yang berlaku iaitu first ialah leading strand and lagging strand. So kita balik nombor satu. Nombor satu apa tadi? Unwinding the double helix strand of DNA. So first step ada 3 enzim yang ada dalam tu masa first step iaitu DNA helicase, single stranded binding protein dengan topoisomerase. Okay, yang kedua synthesis of RNA primer. Enzim yang terlibat ialah enzim DNA primase. Third step
DNA ke RNA?
RNA. RNA primer. Okay. Third step is enzim yang terlibat ialah DNA polymerase 3. And then fourth step ni just apa yang dia akan membentuk leading strand dengan lagging strand. Yang ada dua ni. Ni yang hijau ni ialah leading. Yang ni yang satu lagi ni dia lagging strand akan membentuk Okazaki fragment. Okay. The fifth step is removing the primer. Yang dia tambah tadi primer masa dia dekat step 2 tu dia tambah, and then dekat step 5 ni dia akan buang balik primer tu. Dia akan remove the primer. So enzim apa yang akan remove the primer is DNA polymerase 1. So DNA polymerase 1 remove the RNA primer, and then bila RNA primer ni dah remove, so dia akan kosong, dia akan ada gap kat sini. So apa yang DNA polymerase buat, dia akan fill the gap with DNA nucleotide as well as any gap between Okazaki fragment. This is Okazaki fragment, and then this is RNA primer. Then in the fifth step, DNA polymerase 1 will remove RNA primer and then fill the gap with the new DNA nucleotide and join with the Okazaki fragment. So that is the fifth step, and the last step is joining the Okazaki fragment. Okay, enzim yang terlibat dalam Okazaki fragment ialah DNA ligase. Okay, DNA ligase ni function dia joins the Okazaki fragment by forming phosphodiester bond to form continuous DNA strands. Yang ini dekat lagging strand ni. So DNA ligase ni kerja dia akan join the Okazaki fragment by forming phosphodiester bond to form continuous DNA strands, and then hasil dia akan menghasilkan 2 identical copies of DNA is produced. Understand? Hello.
Okay, Farid.
Understand.
Kai, mana Kai?
Cikgu, saya nak tunjuk video boleh?
Boleh carry on, okay?
Okay, kejap Sebentar.
Cikgu,
Ya.
kita orang ada buat quiz juga tau.
Okay, good.
Kai buat.
Kai yang buat.
Alright, good.
Rakan-rakan nampak kan video ni?
Boleh. Jalan.
Nampak.
Nampak Abang Farid.
Okay, kejap saya besarkan full screen. Kejap je. Awak lama ni. Maaf ya, lag sikit. Lag kan? Sekejap
Nak off spot ke?
Tak, bukan je lag kejap. Okay, sorry.
Farid seorang sebut. Dira, Jannah, Kai, Farah, Farid. Seorang mana? Jannah, Farid.
We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus.
Dengar kan?
Still, both prokaryotic and
Cun.
eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players.
Farid? Farid, are you okay? Your video suddenly stopped. Farid? Okay, class. While waiting for Farid to come back, okay, so teacher would like you to, okay, first of all, congratulations for group Jannah, okay, Farid, Kai, Dira, okay, Farid, Farah, Kai, Jannah and Dira for the good effort, okay, that you prepare to sharing with us about the first subtopic under chapter 6, okay, which is DNA replication. Okay, it's a good effort, and overall for teacher, yeah, quite good. Okay, it's a very good effort. Okay, whatever all the points that supposed to have, semua ada. Okay, and you prepare the good video. Cuma nanti when it comes to writing, banyak lagi yang cikgu rasa you all boleh upgrade. Okay, so actually teacher already assign one simple exercise. Okay, after group Farid have their present their points. Okay, actually everybody you all boleh try to work out. Okay, so let's identify. Okay, just now Farid. Okay, can you see my slides, class? Okay, teacher share the desktop. Can you see my slide? So
Boleh teacher.
okay. So this is what just now Farid and the gang, okay, Farid, Farah, Kai, Jannah and Dira show you the DNA strand that is unwinding. Okay, so teacher would like you to have just now Jannah already Jannah start with this what to say?
Enzyme.
Yeah, enzyme the one protein. Okay.
Function. Oh yeah, okay.
So basically nanti teacher nak you fill up your GC because actually in your GC teacher dah assign you. Okay. So this is exercise that teacher already assigned you in the GC. Okay, so like what Jannah have already started just now. Okay, you need to understand the function of each enzyme or protein that involved in the DNA replication. Okay, so next after that, teacher also share with you the explanation. Okay, if you refer to our notes, there are 6 steps. Okay, so teacher would like you nanti, okay, slowly fill up this the exercise, okay, we're using the correct answer, okay, to describe the DNA replication process. Right, okay, so you can use this, and teacher would like, okay, just now teacher would like to emphasize this now class there are 3 types of model, okay? But scientists agree that we follow which model, class? Okay, the model we follow is a semi-conservative model. Okay, we follow the semi-conservative model. Okay, so as you all can see here, okay, the model replication we follow the semi-conservative model. Okay, where it involve one parental DNA, okay, and duplicate to become new DNA strand. Okay, another parental DNA replicate to become new DNA strand. Okay, so this is how it works. And always remember, class, okay, new DNA must produce from 5 prime to 3 prime. Okay, 5 prime to 3 prime. Okay, so meaning if this one is 5 prime to 3 prime, okay, so the new DNA that produced is, okay, class, this is the new DNA produced, 5 prime to 3 prime. Okay, so this is a parental 3 prime to 5 prime. So the new DNA that produced will be 5 prime to 3 prime. That is how it works. Okay, right? So now, okay, nanti you refer to the diagram class that teacher already assigned in your Google Classroom. Okay, so this is what teacher now you all have an idea about it. Okay, like just now Jana mentioned leading strand, okay, lagging strand. Farid also mentioned class, okay, position of 5 prime to 3 prime. Okay, so you have 5 prime to 3 prime. So this is a parental. This one we call as a parental strand. Okay, so just now group Farid juga mention about this one, origin of replication. Okay, origin of replication. Okay, this is origin of replication. Okay, and teacher would like you to have an idea for eukaryote. We have multiple origin replication. Eukaryote, like us, like animal, like human. Okay, just imagine we have 46 chromosome. Okay, 46 chromosome that must duplicate or that synthesize to the new strand. So that's why when you learn during chapter 3, okay, under synthesis process, okay, interface 3 phases. So the longest phase under interface is replication. Why? So now you understand why. Okay. So like teacher mentioned just now, class, this is leading strand. So where is the replication fork? So this is the replication fork. Okay, replication fork. And teacher kotak ni, this is replication fork. Right, so like teacher just now mentioned, production of new strand must from 5 to 3. Okay, so this 5 to 3. Okay, 5 to 3. Okay, same go like this. This is 5 to 3. 5 to 3, 5 to 3. Okay, so this also 5. Okay, 5 to 3. Okay, this one is 5. Okay, 5 to 3. Okay, so what is this one, class? The red one, this one. Okay, this is what Farid mentioned just now. This is RNA primer. Okay, Jana, again, apa function RNA primer just now? RNA primer?
RNA primer.
What is the function of RNA primer? RNA primer?
Catalyze, synthesize the short RNA.
Yes, okay. Catalyze the short
RNA primer.
Okay, primase. So how to produce RNA primer is catalyzed by the RNA primase.
Primer.
Okay. So position 5T. Understand, guys? So in this case, this is bidirectional from origin of replication. This is origin of replication, so bidirectional. Okay, so bidirectional. Okay, so it's a bidirectional. Right, so now let's label this diagram, okay, in order you to understand better. Okay, so Farah, Farid, Kai, Jannah and Dira, you must assist your friends, okay. So because you are the one that doing this topic. Okay, so let's label the topic that given here. Everybody nanti you all sign apa ni, refer to your GC teacher dah assign this question in your GC, nanti teacher nak you turn in nanti, okay? So just now, okay, Jannah already introduced us to all the enzyme and one protein, SSBP. So let's identify the position of all the enzyme in this DNA strand. Okay, let's start with A. What do you think is A, Jannah? A, do you think A and D are similar? A and D, both
Yes.
sides, both strands we have
Sama.
we found it. So A and D will be?
Sama.
Yes. So what is A and D? DNA? DNA polymerase?
3.
Yes, DNA polymerase 3. So this is DNA polymerase 3. A and D somewhere. Okay, then this is the most important class, unwinding. Unwinding the DNA strand. We need E. So what is E, Khai? We need this enzyme E.
Salah.
This enzyme E. Yes, so E is helicase. Right? Okay, then like Farid mentioned just now. We need this enzyme, this enzyme C. Okay, this red color is the RNA primer. This one is RNA primer. So the pink molecule supposed to be the C will be apa class? Primase. Okay, primase. Right. So helicase unwinding. Okay. Now, because DNA, as you all know, it's a super coil. So how to release the tension? So we need this structure. Okay, we need this structure. The teacher, yes, teacher label this one. Okay, so this one. Can see we choose another color. Okay, this one. Okay, this one class, this one and this one, both sides. So what is B? B is
Topo.
B is topo.
Isomerase.
B is topo isomerase. Wait, no. Topo isomerase is just C is primase. Okay, good. Okay, B both sides. Nanti kata avoid supercoiling. No, not supercoiling. We want the
Reformation of
we want the just the SSBP. Apa tadi? Jana, what is the function SSBP? What is the function SSBP just now?
Single stranded. Kan
SSBP, yes. SSBP.
Single stranded
Attached
binding
to?
protein.
Attached to each single stranded to class.
Prevent.
SSBP stabilize the two
Unwound single DNA strand.
Untuk prevent them from rewind back.
Rewind back.
Okay, so this is B is SSBP. Short form, single stranded binding proteins, because that's why both sides each single strand. Okay, you find SSBP. Okay, so A done, B done, E done, C done, D done. Okay, next, what is E? This one E. What is E?
DNA helicase.
Okay, and this one, no, E. This one is F, sorry. E done, E or D done, any case,
Polymerase 1.
Yes, DNA polymerase 1. Okay, F is DNA polymerase 1. And this one, G is the ligase.
DNA ligase.
F is a DNA polymerase 1. Okay, so this is actually all the enzyme and all the and also one protein that involve as a apa ni having its specific role, okay, in DNA replication. Okay, Farid, are you in? Let's check.
Yes teacher, sorry. Tadi ada
So
masalah.
your video is it okay? Dah boleh run ke?
Alamak, dah terpadam lah teacher. Tapi
Okay,
saya
next
ada, sekejap, saya ada dekat file, saya dah download dah video tu.
Okay,
Wait
nanti
apalah.
Farid share in our Telegram lah.
Okay.
Cikgu,
Ya.
kita
Cikgu.
nak buat kuis sekarang. Sebab kuis
okay
tu
lah.
live,
So-
semua kena jawab sekaligus.
Alright, so now because their group already ready for quiz, so teacher apa ni stop sharing and allow their group to share the question. Okay, so alright, so teacher pass to Jana's group.
Okay.
Buka grup bio.
Kita bagi link. Semua masuk ya.
Semua masuk je dekat quizzes tu.
Oh, quiz it, okay.
Farid pun masuk.
Okay.
Aku tengok 4%.
Dah siap?
Kat sini.
Participate semua kelas. Nanti teacher akan ambil rekod ni sebagai apa tu your performance because your understanding about the DNA replication that have been shared by Farid's group.
Sekejap je, saya share. Ya, saya dah share dekat dalam ni. Ada boleh tengok.
Okay.
Teacher.
Yes. Wah, itu Farid punya.
Ya, teacher.
Wah, Bihang dah siap?
Zaki. Ini.
Wow, power up.
Hehe. Finding nucleotide. Pelan.
Apa soalan ni?
Ada 15.
Okay. Ini soalan ke-7.